Hello, my name is Pawel, and I will be talking about leveraging massive protein structure datasets for function prediction. Due to recent breakthroughs from AlphaFold and ESMFold, we have access to a huge number of high-quality structure models. Tools such as DeepFry show that utilization of such information can lead to much better function prediction as compared to sequence-based methods. We can further use metagenomic DeepFry, which is an extension of DeepFry, to map sequences to putative structures and have full capabilities of structure-based DeepFry. DeepFry 1, the original DeepFry, produces high coverage annotations. However, they are slightly more general as compared to homology-based methods. Here, in my talk, I will show how DeepFry retrained on AlphaFold UniProt dataset can elevate this limitation. So we use two inputs, gene ontology annotations of UniProt, where each Go term needs to have at least one experimental annotation to be considered, and high-quality predictions from AlphaFold. And then each structure is annotated by Go term vector. We also use Go hierarchy propagation to increase number of annotations. So thanks to that, almost 3 million of structures are annotated by almost 60 million of annotations. And then we use this as a training set. We also use the original DeepFry 1 architecture, which has been proven to be reliable. And the final DeepFry 1.1 can predict almost 9,000 of Go terms, which is more than 3 times more as compared to original DeepFry. If we have a structure, we can directly use DeepFry 1.1 for function prediction. On the other hand, if we have sequence, we can use metagenomic DeepFry to find the closest homolog, and then use DeepFry 1.1. The upgraded version allows not only for a larger number of predicted Go terms, but they are also more informative. And informativeness of a Go term here is defined as minus log probability of finding a given Go term in genomic database. DeepFry produces a score for each Go term, so it is crucial to know which value is reliable. Here I present precision against DeepFry score for both versions using the structures from protein databank. So at least when working with structures from the dataset, we should use score at least 0.35 for V1 and at least 0.5 for V11. And the difference, the worst performance for the latter, is due to the fact that DeepFry 1 has been trained and tested on the same distribution, namely PDB and related structures, whereas 1.1 has been trained on UniProt and tested on PDB. So we compared both versions on MIP dataset, so Microbiome Immunity Project dataset, that comprises relatively short single-domain structure models from GEBA dataset. And as we can see, V11 allows for a higher number of very informative Go terms as compared to the original DeepFry. We also mapped Go terms into categories, and again we can notice that V11 allows for much higher coverage of categories. Interestingly, those green categories from information storage and processing in V1 are predicted by DeepFry 1.1 as also green categories, and also this cell cycle control, cell division, and chromosome partitioning, which is somehow related. We also found some high-quality predictions of very hard targets, so the putative novel faults from MIP database. As we can see, the DeepFry score is very high, and at the same time, the information content is also very high, between 12 and 14. We also ran DeepFry 1.1 on random pool of almost 1 million of predictions from ESM Atlas. So as we can see, we can notice high coverage across wide IC range. We also compared DeepFry against Pro10 on that dataset. On the DeepFry side, we use only high-quality annotations, excluding general Go terms. On the Pro10 side, we use all BFAM annotations. And as we can see, we have more annotations, more annotated proteins as compared to Pro10. More results and details will be presented during my talk at Function Cozy on Wednesday. So to sum up, the upgraded DeepFry provides not only more Go terms, but they are also more informative. We can use metagenomic DeepFry to extend DeepFry functionality to predict functions for practically any sequence dataset. In the future, we plan to upgrade DeepFry even more, so stay tuned. Thank you very much for your attention.